In this chapter, we are going to be covering cells and organelles. So what we're going to start with is the cell membrane. So as we see here, this is the cell membrane. And what this does is it contains things such as phospholipids, cholesterol, and proteins. So we're going to start with phospholipids. So we're going to see that it has a hydrophilic head, so it loves H2O, and a hydrophobic tail. So what happens is that they're going to arrange themselves that both hydrophilic ends are towards water. So now if we look at the head and then we look at the tail, what we find is that we have a 3-carbon, notice the carbon here, and here, and here, 3-carbon backbone. So now what we have is the head and then the 2 tails, right? But it seems like there's 2 heads, but really it's just 1, right? And then we have a phosphate group attached to it. So it's getting a little bit messy here, but if we have this, and then inside we have the CH2 right here attached to the O, the phosphate group. So basically you still have the 3-carbon backbone, except it's just with 1 head with a phosphate group. Notice that the phosphate group is polar because it has the negative charge. Also notice how the fatty acids are nonpolar, so they are hydrophobic. Now due to it being both hydrophilic and hydrophobic, we call this amphipathic. So you're going to write down amphipathic. Oops. So if you recall how in the RNA world hypothesis they spontaneously assemble, this is an example of it, and this is why. When placed in an aqueous environment, they will arrange themselves in this manner. Next we have something called cholesterol. So if we look at cholesterol, we have 4 hydrocarbon rings that benefit the phospholipid bilayer in terms of fluidity. And we'll take a look at this right now. So in our cell membrane, we have phospholipids, cholesterol, and the third thing that we have is membrane proteins. So we have 2 types of membrane proteins. We have membrane proteins throughout the whole entire phospholipid bilayer for both sides. It comes in and out. And we also have a peripheral membrane protein, which is just on the external part. So what happens if you want to bring a polar molecule through the phospholipid bilayer? Well, it would have to go through the integral protein. And why can't it go out without the integral protein? Well, it's because the phospholipid bilayer, remember, it has the hydrophobic tail. So because of this, you need to have something push it through to get it across the cell membrane. So largely, integral proteins are mostly just for transport. So recognition proteins are attached to polysaccharides called glycoproteins. So what they do is they identify each other when they come in contact. Adhesion proteins attach to other things like cells or anchor themselves into the cytoskeleton. Hence the name adhesion. Receptor proteins actually receive chemical signals from the outside environment, and instead of inducing a response themselves, they carry on a secondary response within the cells. So they tell another thing in the cell to cause a response. Essentially, our cellular membrane is made up of 3 things: the phospholipids, cholesterol, and membrane proteins. This all together creates something that we call the fluid mosaic model. So now we're going to talk a little bit more about fluidity. So specifically, cholesterol helps phospholipids stay packed together in hot conditions and in cold conditions. So it keeps it flexible in hot conditions because when the phospholipids separate, then they tend to want to connect to the cholesterol, so they kind of come towards each other. Now, in cold environments, it keeps the cellular membrane flexible and fluid because it maintains some distance between the phospholipids. Now, if we're going to delve deeper into fatty acids, we have saturated and unsaturated fatty acids. So when you have saturated, that means you have the most amount of hydrogens possible. Therefore, it's all single bonds. There's no double bonds. But if it's unsaturated, that means you're missing hydrogens and that you do have double bonds. So if you have a cis unsaturated fat, then it stays going in the same direction. And for trans, what happens is that they go on opposite sides of the double bond. They pack together extremely tightly, while in the cis, there are kinks. So now what happens is that if you have a double bond or an unsaturated fat, your phospholipid is going to look wonky like this.